So let's talk about the electron transport chain. The electron transport chain is also known as oxidative phosphorylation. And before I tell you the meaning of these two terms, I want you to understand the whole process so that you can come up with your own definition. Now, what's the main function of the electron transport chain? It's to produce molecules of ATP, energy. And how does it do this? It requires two molecules named NADH and FADH2. And how do we yield these molecules? I want to dive deep into one of the metabolic processes to make you understand this. So we all know that glucose can be converted into pyruvate through the process of glycolysis. Now, I want you to understand that the process of glycolysis occurs in the cytoplasm of the cell. And I'll tell you why this is important later. But through the process of glycolysis, we can yield two molecules of NADH. Further, pyruvate can take one of the four metabolic pathways. One certain pathway can lead to the formation of acetyl-CoA. Now we know that acetyl-CoA is a major component of the TCA cycle. And here I want you to remember that the TCA cycle occurs in the mitochondria. I'll tell you exactly where in the mitochondria a bit later, but right now I just want you to know that it occurs in the mitochondria and glycolysis occurs in the cytoplasm. And how much NADH and FADH2 does the TCA cycle yield? It yields us 6 molecules of NADH and 2 molecules of FADH2. Now, I did tell you that the TCA cycle occurs in the mitochondria, but where exactly? Now, the mitochondria has an outer mitochondrial membrane, the inner mitochondrial membrane, and between the two membranes, the space between it is actually called the intermembrane space. And the innermost part is called the matrix. And the matrix is where the TCA cycle occurs. And one more thing that I need you to know is the entire electron transport chain is actually embedded on the inner mitochondrial membrane. So let's zoom in to the inner mitochondrial membrane. I know this looks like a lot, but trust me, I'll make it a lot easier. So the electron transport chain is embedded on the inner mitochondrial membrane, this whole space. And it has 4 complexes, complex 1, 2, 3, 4, and 2 molecules called coenzyme Q and cytochrome C. Along with this, this whole thing is actually called the electron transport chain, and it has an additional particle called the F1, F0 particle, or you can also call it the ADP synthase. Now, as I said earlier, the TCA cycle occurs in the matrix of the mitochondria. And so the entire electron transport chain has access to the NADH and FADH2 that it yields. But what about the NADH molecules that are derived from glycolysis? That enters the mitochondria with the help of a shuttle and reaches the matrix as well. So now that we know where we get our NADH and FADH2 molecules, let's start with the process. So NADH, not FADH2, only NADH, donates its electrons to complex 1 and releases its protons NAD plus and H plus into the matrix. Now, the electron donated undergoes multiple redox reactions in the complex 1. and complex 1 gets supercharged. And because it's now supercharged, the H plus ions, which are present in the matrix, get shooted out of the complex 1 and enters the intermembrane space. Okay, so now there's a lot more H plus ions in the intermembrane space compared to the matrix. And that forms a proton gradient. Now, once this is done, the electron is transported to coenzyme Q. Now, FADH2 donates its electrons to complex 2 and releases its protons FAD plus and H plus into the matrix. Again, this electron undergoes multiple redox reactions and supercharges complex 2. Unlike complex 1, complex 2 can't move protons from the matrix into the intermembrane space. So instead, it sends its electrons to coenzyme Q. Now, coenzyme Q has electrons from both complex 1 and complex 2. Once it's collected the electrons, coenzyme Q passes down the electrons to complex 3. Again, the electrons undergo multiple redox reactions and supercharge complex 3. And once the complex gets supercharged, the protons present in the matrix are shooted out into the intermembrane space, again increasing the proton gradient. Now, I want you to know that whenever a molecule like NADH or FADH2 donates an electron, it's undergoing oxidation. And I just want you to keep this in mind because it will help me explain you what oxidative phosphorylation is. So now, once the electrons have been shooted into the intermembrane space, the electrons are then passed on to cytochrome C. And cytochrome C then later passes these electrons to complex 4. Again, the electron undergoes multiple redox reactions and gets supercharged, and once it's supercharged, it removes the protons from the matrix into the intermembrane space. Now the question is, where does this electron go? The electron finally exits the entire complex system, enters the matrix, and oxygen accepts this electron as the final electron acceptor. And oxygen then splits into 2 oxygen ions, and the free-floating hydrogen protons combine with these oxygen ions to form 2 molecules of water. Now, this is the whole transport of electrons. But where is the ATP produced? As we've seen, all the hydrogen protons are now in the intermembrane space. So the final step is that because we know that molecules tend to flow from a higher concentration to a lower concentration, these hydrogen ions actually flow through the F1 particle. And while the ions actually flow through F1, it kind of spins this particle like a wind turbine and produces energy. So the hydrogen atoms actually re-enter the matrix, but with the energy produced, ADP molecules join with phosphate ions and form ATP. And this is the whole process of oxidative phosphorylation. So with that energy, ADP joins with phosphate and through the process of phosphorylation forms ATP. And as I said, phosphorylation occurs here, and we've seen a couple of oxidative reactions here. So this process together is called oxidative phosphorylation, and that's where the term comes from. Now, the last thing that you need to know about this topic is the name of the complexes and the uncouplers and inhibitors. So complex 1 is known as NADH-CoQ reductase. Complex 2 is known as succinate CoQ reductase. Complex 3 is known as cytochrome C reductase. Complex 4 is known as cytochrome C oxidase. And the F1F0 particle is also known as ATP synthase. Now, let's talk about the inhibitors. The complex 1 is inhibited by rotenone and phenobarbital. Complex 2 is inhibited by malonate. Complex 3 by fenformin. Complex 4 by carbon monoxide, cyanide, and hydrogen sulfide. And ADP synthase by oligomycin. And the last thing that you need to know is about the uncouplers. Uncouplers are those which disrupt the oxidative phosphorylation. It uncouples that process. And one of the drugs that uncouple this process is 2,4-DNP. And two physiological factors that uncouple this process is called thermogenin and thyroxine. And that's it.